For part 2, we're going to start with anatomy of the stomach. So our food bolus has made its way to the stomach. Through the, we just have the cardiac opening. So location of the lower esophageal sphincter is here. This is known as the cardiac region or cardia of the stomach. So some resources are going to refer to this as the cardiac sphincter. They're both perfectly accurate. If you're looking actually into an abdominal cavity, let's say you've got this sort of anterior but also inferior view into the abdominal cavity towards that respiratory diaphragm, and the stomach is here. If you're looking up towards that diaphragm, you're going to be able to see the heartbeat on the other side of that diaphragm, that respiratory diaphragm, because the mediastinum is just on the other side of that respiratory diaphragm. The location of the heart is just superior to this structure here. So the heart's going to be beating very, very physically close to this. You're going to see that pulsing movement right here if you happen to be doing some surgery in the abdominal cavity. That's what that's going to look like. There is a general term here known as the fundus. A fundus, for any anatomical structure, is a superior domed region. So this happens to be the fundus of the stomach. Superior domed region of the stomach is the fundus of the stomach. You do have to say fundus of the stomach. If you don't say fundus of the stomach, then you're going to say, somebody's going to say, what fundus? Which fundus? What fundus are we talking about? Because there's the fundus of the uterus, for example. So you do have to say, be specific here, fundus of the stomach. Unless you happen to be having a conversation exclusively about the digestive system and we're talking about gas, then probably they're not going to be confused about it. But for now, I want you to get used to being specific with your anatomical terminology. You still have the same 4 layers. You still have the mucosa, submucosa, muscularis, and adventitia. Your muscularis layer has an additional oblique muscular layer. So you would call this mucosa. There is a submucosa that I don't think, yeah, they've illustrated it, kind of. Sure, why not? That layer in there, but on the surface of that submucosa, we do have our oblique muscular layer, but then we have our good old familiar circular muscular layer and longitudinal muscular layer. And then that outermost surface of the stomach, you can reach into the abdominal cavity and touch it if you're dissecting. That's a serosa, as we know from our discussion about serosa versus adventitia earlier. Let's see, the body of the stomach, again, this is the cardiac region. This is the pyloric region, or pylorus. So this is the pyloric sphincter, and I'm grateful that this image is calling it a sphincter. Some resources say it's a valve. There's nothing valvular about it. It doesn't do this. It does this. So it's a sphincter, not a valve, and this has it correct, which makes me happy. Then you go into your first portion of small intestine for there, which is the duodenum. So the duodenum is most closely associated with the stomach of all the 3 parts of the small intestine. The duodenum is the proximal small intestine. I have heard other people refer to it as the duodenum. You may hear the phrase duodenal ulcer. That is referring to the same structure. Did I miss anything? Oh, lesser curvature, greater curvature. I think those are pretty intuitive. Also, rugae. So I've used the word rugosity to refer to bony structures before. Bony features can be more or less rugose. That means how roughened they are. Now, when you have not eaten Thanksgiving dinner, your stomach is empty, you're in a fasting state, your stomach is not distended and it's relaxed, you will have very deep, dense rugae. They'll be very folded. But if you do eat Thanksgiving dinner, if you eat a very, very large meal, then your stomach becomes distended and those rugae kind of stretch out and become smoothed because of consuming large volumes of food. That's it. Now, within those rugae of the stomach, histologically, when we start getting to a more microscopic level, we do have what's known as gastric pits with gastric glands. So this is a simple columnar epithelium, but it's highly invaginated. It's folded in many, many times. Got something on my eye and it's very itchy. Okay, there we go. So here's your gastric pit, and deep inside of this gastric pit, we do have some very specialized cells. They are parietal cells, chief cells, and some very specific endocrine cells. I've seen these with a couple of different names. Endocrine cell is fine. We'll stick with that. Parietal cells and chief cells, those names come back again and again. You absolutely need those. We'll deal with that later. Let's see. Again, as I mentioned, you have the same general layers. This is the mucosa. This is the submucosa. This is the muscularis. Now we have 3 layers of muscularis. And the serosa here. Now let's look at what these cells are doing. Parietal cells and chief cells in particular, but we do have to look at endocrine cells as well. Here's that histology. So you may remember this. Did we do a lot with histology in here? I don't think we may have done this in histology in here. We looked at simple columnar, but I don't think we looked at any real slides for simple columnar, especially not any that are as difficult for a novice to read as a gastric pit. So this is actually a pretty typical epithelium to use in BIO127 for a simple columnar epithelium, even though it is this folded. It is a very reliable source for a simple columnar epithelium. And the most important part of pattern recognition for this is understanding that we are looking at a gastric pit. It is a simple columnar epithelium, it's just very folded. So deep down here in the gastric pit is the glandular cells that make this a gastric gland. And of course we do have multiple cell populations down here. I would never ask you to differentiate a chief versus parietal cell in histology like this at this level. But you do need to know that they're both there, and knowing a little bit about what they look like can actually help you determine what they're releasing, because parietal cells are actually going to release hydrochloric acid. You can see this little clear portion in it, because that's just a fluid. Chief cells stain a lot darker because they are producing a protein known as pepsinogen. So again, that's what's going to contribute to the chemical breakdown that starts in the stomach. So the stomach is squeezing in multiple dimensions with its 3 layers of muscularis. So it's doing the mechanical breakdown. Most of that is about exposing new surfaces of that food bolus to these digestive enzymes at this point. So you did a lot of mechanical breakdown in your mouth, and now you're breaking it into different pieces, exposing new surfaces to hydrochloric acid, to pepsinogen. Let's see, I need to move myself. Faster regulation of delivery. I'll move myself over. Here we go. Nicely done. Okay. So we have the start of chemical breakdown of especially proteins in the stomach. Remember, the salivary amylase started carbohydrates in the mouth, but pepsinogen is, when it combines with hydrochloric acid, is going to help start break down proteins into smaller units. We'll also have mucus protecting the stomach lining from the acid, because guess what? The human body is not a huge fan of this acidic of an environment. Everything in your stomach has evolved to work in that situation, so pepsinogen specifically works in the presence of hydrochloric acid, which is not typical of something like catalase, for example, as you know. Okay, so I've been calling it the food bolus so far. When we went from here to here, that was a food bolus. Once it's out of the stomach and it's mixed with some enzymes and it's mixed with some hydrochloric acid and some mucus, it's chyme now. It's no longer a food bolus, it's chyme. That chyme entering the duodenum is the thing that starts to alter a hormonal milieu. So we're going to have new hormones released when food makes it into the duodenum. Your stomach doesn't just dump everything into the duodenum. Your stomach fills with food, and then a certain amount, that pyloric sphincter opens and allows a certain amount into the duodenum, and then another certain amount, and another certain amount gradually. Gradually is what we want. We don't want it all dumped. There's actually something called dumping syndrome, which is where the pylorus of the stomach dumps everything into the duodenum, and it's really unhealthy when that happens. So we do have this sort of rhythmic contraction in our three muscular layers, allowing for the movement of food chyme now to make it into the duodenum. So we have the opening and closing of the pyloric sphincter. I think that's all I really want you to get out of that. Now that it's in the small intestine, a new set of things has to happen. We do have to stop activity in the stomach. Stomach's done. Time for something else. But we have to start some things in the small intestine. And that's really why the duodenum is different from the rest of the small intestine, is it actually has some pretty important endocrine functions. When we get to the endocrine system, we are going to find that there are a lot of roles to play here. I forget what I was going to say about that. I'll come back to it. Anyway, we're in the duodenum. And, oh yeah. So you can have an organ, and that organ can have its role, right? It can also have a secondary role as an endocrine organ. So for example, the duodenum is going to release something called cholecystokinin, CCK, which we're about to play with. Your kidneys do what kidneys do. They make urine, but they also have an endocrine role, synthesizing something called EPO and also something known as renin. So different organs have multiple roles. You also have things that are glands that are only endocrine organs, and they don't do anything else. So we consider that a secondary role of the duodenum, for example. It's a passageway for food, but it's also an endocrine organ. So once we get to that duodenum, in addition to transferring that food to the next part of the small intestine, it's also going to release some important substances. Let's see. Now, the small intestine as a whole, it does produce some digestive enzymes, so we are still digesting with enzymes, chemical digestion. We're going to break down the rest of the macromolecules of life, or break down the things that are already there further, right? So we already had salivary amylase that started in your mouth, that broke down some carbs. We had pepsinogen that started in your stomach, breakdown of certain proteins. But we still are going to release things that help specifically with fats, specifically for carbs, specifically for proteins, and nucleic acids as well. We're not going to focus as much on those. Most absorption is happening in the small intestine. Not really the large intestine. The small intestine is actually the rock star for absorption. We're also going to produce buffers. Remember, we had some bicarb in the saliva. We're going to have something similar happen in the duodenum because, remember, the stomach was a very acidic environment. You secreted hydrochloric acid, and that is not safe for the rest of the gut tube. So we're actually going to produce some buffers and release them actually into the duodenum specifically to neutralize that chyme that came out of the stomach. This is in order: duodenum, jejunum, ileum. I have a little ditty if you're ready for it. Duodenum, do 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 do. Jejunum, do 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 do. The ileum, do 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 do. I hope that helped. Okay. Now, what makes the jejunum different from the ileum? The duodenum's pretty unique. It's in a specific location. It's pretty short, and it has this endocrine function, so it's distinct. Anatomically, physically, histologically, everything is distinct about the duodenum. But the jejunum and the ileum are the rest of the small intestine, and because the rest of it is very wobbly, it's going in all these different directions and circling back on itself, how do we tell if it's jejunum or ileum? The mucosa of the small intestine has various levels of folding. In fact, let's go a little bit further. So you have these folds. They have some different names. One of them is plicae circularis, but I think we're just sticking with intestinal folds right now. Your jejunum is very, very, very foldy. On these folds, throughout the small intestine, you do have finger-like projections known as villi. So it's folded once, but then the tissue on these folds is itself folded. And then if you were going to go even smaller and look at the microscopy of this and look at this histologically, each of these villi, so we're just looking at one of these and zooming in on it, every single one of these villi has those simple columnar cells, which themselves have microvilli. So we have folds on folds on folds. And why do you ever have folds? You have folds to increase surface area. You always have folds to increase surface area. So this is 3 orders of magnitude of increasing surface area. This is why 95% of your food is absorbed in your small intestine, and that's also what's different between the jejunum and the ileum, is these intestinal folds are much more prominent in the jejunum than they are in the ileum. So you're getting most of your absorption actually in the jejunum. And then that still happens in the ileum, it's just slightly decreased. You'll still have intestinal villi and you'll still have microvilli in the ileum, just the folds get a little less structured. Here's the wall of the duodenum, again with the actual villi themselves. So remember, we had the stomach histology with gastric pits and gastric glands. We have crypts down here, but we don't have our chief and parietal cells like we did in the stomach. So we still have some interesting glands down here. Let's see, what else do I want to show you? I can show you goblet cells again. So those are, maybe I haven't shown you goblet cells before. It's hard to do all this prep work when I'm doing A&P and BIO127 at the same time, because it's a lot of overlapping content, but not always. So you have these special cells that are just producing mucus, and they'll be solid at the bottom of the cell, but the rest of the cell is shaped like a goblet, and it just encompasses this little blob of mucus. And you can see that in histology, and that's what those are. So they're just making mucus. All this mucus will be released into that mucosa right there, and into that lumen. I think that's all we're doing there. Yeah, it's time to move on, so that's a good stopping point. We got through the stomach and small intestine. Yeah. Next we'll do some other structures, including enzymes.